How do prostate cancer cells change at the molecular level? Imagine a tiny change inside a cell that can lead to big problems later on. That's what happens in prostate cancer cells at the molecular level. These cells go through a series of changes in their genes and how they control their activities. These changes help the cancer grow, spread, and sometimes resist treatments. Understanding these changes can help with early detection and better management of prostate cancer. One of the main ways prostate cancer cells change is through genetic mutations. Genes are like instructions for cells, and mutations are mistakes in these instructions. For example, mutations in genes called TP53 and PTN are common. TP53 helps control cell damage and can trigger cell death if something goes wrong. When TP53 is mutated, damaged cells don't die as they should, which can lead to more aggressive cancer. PTN normally acts as a brake on cell growth, so when it's lost or mutated, cells grow uncontrollably. These mutations are more common in advanced prostate cancer and are linked to worse outcomes. Another important change involves the androgen receptor gene. Prostate cells depend on androgens, which are male hormones, to grow. In early prostate cancer, these cells need androgens to survive. But as the cancer progresses, especially in castration-resistant prostate cancer, the cells adapt. They can mutate the androgen receptor or produce versions that stay active even without hormones. This makes the cancer resistant to hormone therapies that lower androgen levels. Changes in DNA repair genes like BRCA1, BRCA2, and ATM also play a role. When these genes are mutated, the cell's ability to fix DNA damage is reduced. This leads to more genetic instability, which can drive cancer progression and make the disease more aggressive. Additionally, gene fusions involving ETS family transcription factors, such as TMPRSS2ERG, are common. These fusions change how genes are regulated, promoting tumor growth. Beyond genes, prostate cancer cells also undergo epigenetic changes. These are modifications that affect gene activity without changing the DNA sequence. For example, abnormal DNA methylation can silence tumor suppressor genes or turn on oncogenes, helping the cancer survive. MicroRNAs are small RNA molecules that regulate gene expression. In prostate cancer, some microRNAs like miR-379 are decreased, which can reduce cell adhesion and allow cancer cells to spread more easily, especially to bones. Signaling pathways inside the cell also become dysregulated. The androgen receptor pathway is central. Early on, cancer cells relying on androgens, but later they can mutate the receptor or produce variants that stay active without hormones. This makes the cancer resistant to hormone therapy. Other pathways like the pi3 degrees Kelvin acadium-TOR pathway often become overactive due to PTN loss, promoting cell survival and growth. Changes in Wnt signaling and amplification of the MYC oncogene further support tumor growth. When tumor suppressors like RB1 are lost or mutated, the cancer can become more aggressive and change its characteristics, sometimes turning into a highly resistant form called neuroendocrine prostate cancer. Finally, metabolic changes occur. Advanced prostate cancer cells often increase their use of glutamine, an amino acid that fuels rapid growth and helps them resist therapy. In summary, prostate cancer cells change at the molecular level through a complex mix of gene mutations, epigenetic shifts, altered gene activity, and disrupted signaling pathways. These changes help the cancer grow, spread, and sometimes resist treatment. Recognizing these molecular changes is crucial for early detection and developing targeted therapies. Always remember, each person's cancer can be different, so personalized medical advice from healthcare professionals is essential.